Good morning, everyone, and welcome to this discussion of the important COVID-19 research being done at the Gladstone Institutes. I'm Bill Younger, President of the Gladstone Foundation. With me is Dr. Deepak Srivastava, President of the Gladstone Institutes. In addition to leading the Gladstone, Deepak is a world-class scientist and clinician. Under his leadership, he's attracted a number of eminent scientists who have pivoted to COVID-19 research. One example is Jennifer Doudna, the inventor of CRISPR technology, and in the minds of many, likely to be Gladstone's second Nobel Prize winner for her discovery. Just recently, Deepak organized two new Gladstone Institutes. Melanie Ott has been named director of the expanding Virology Institute, and Alex Marson is director of the new Gladstone UCSF Institute of Genomic Immunology. All these people are building on a long, outstanding record of important discoveries in virology, especially HIV/AIDS, by Gladstone investigators. I'd now like to turn the program over to Deepak.
Thank you, Bill, and welcome everybody to this webinar. Thank you all for spending a little time with us today, and I hope all of your families have been okay through this period. And I've said to many of you, I think we have mostly our Gladstone family here on the phone on the call today. If any of you or your family members run into any trouble during this period, please do feel like you can contact me or others here at Gladstone to help you navigate during this period. And hopefully none of you will need that, but if so, we are here to help you with that. As Bill mentioned, we are at a very exciting time in Gladstone's history right now. Many of you know we've had a virology and immunology focused institute since 1991 that we created to focus on HIV/AIDS at that time, at the height of the epidemic, and with San Francisco being the epicenter of that global epidemic. And that group of virologists over the years transformed HIV from a uniformly lethal disease to what is now a chronic disease, not yet cured, and we're still working on that. And Warner Green led that institute for nearly 3 decades, and with great international prominence and great contributions to that field. And even before the pandemic, we had initiated a search about a year ago to find a new director for that institute, and we had underwent an international search. And it turned out that as we went through that search, it became clear that virology and immunology, which we had housed in one institute, while they intersect quite deeply with one another, really had moved over time to a point where if we wanted to be world leaders in both areas, that it might take unique expertise in both virology and immunology. And that's what led to us announcing, as many of you might have seen a week or two ago, the creation of 2 Gladstone Institutes, the Institute of Virology and of Genomic Immunology. And both of those areas have tremendous potential to impact human health in many ways in the coming decades. And so Gladstone is going to make a significant investment in both those areas. The virology aspect is, I think, clear, and we'll talk more about that today, how it's become more clear to, I think, the whole world, how important those efforts are, but it's been part of our DNA for a long time. And the ability to manipulate the immune system for so many diseases, including cancer, which all of you are probably familiar with, but also for infectious diseases, for autoimmune diseases, and inflammatory conditions, has tremendous potential. And Alex Marson, who will lead that area, is one of the rising stars in that area. So I wanted just to mention that because we just announced that, and it's really a pivotal moment, but it gives us a tremendous amount of momentum going forward to tackle both of these areas. Now, today, I wanted to share with you both initially a broader landscape of where we are currently with the COVID-19 pandemic, and more specifically, how the investigators at Gladstone are trying to impact this disease and really make a difference. And as a backdrop to all that, I should say that I'm engaged every week at the moment with calls with people in the philanthropic community, the government at the highest levels, and corporations on the commercial side, sharing what we're doing. And I can tell you that universally in the three areas in which we're trying to make an impact that I'll share with you, I repeatedly hear that despite our relatively small size, we are doing some of the most creative work in this area. And I'll try to put that into context for you of what else is going on in the landscape worldwide, which is tremendous. There's a tremendous effort with great collaboration across the world to fight this together. So you should feel good about that in general. So with that, let me share my screen with you. And I'll try to spend a bit of time giving you some, sharing some slides with you and some comments, but leave ample time for questions so we can have a broader discussion around that. All right, and for those of you who want to learn more as we go along, we'll put our URL in the chat room and you can take a look at that later. So maybe before we dive deep into some of the science that is going on in this space, let me just make sure we're all on the same page regarding the virus that we're dealing with. So coronavirus, as I imagine most of you have learned by now, are not something new. They are actually a common virus. About a third of the colds that we all have every year are from a type of coronavirus that is not so serious. But there have been now 3 very significant coronavirus infections across the world in this century alone. And those include SARS and MERS, which you're probably familiar with. And those gained a lot of attention. But if you look at the number of infected, it was under 10,000 for both of those. And the number of deaths were under 1,000 for both of those worldwide. Yet those are in everybody's mind because they did get a lot of attention and concern. In contrast, the COVID-19 has affected now, as of today, over 5 million people that are documented positive. There are far more that are positive, I'm sure, and over 300,000 deaths. And about 30% of both of those numbers are from the United States alone. So as you, I'm sure know, we're approaching rapidly 100,000 deaths in this country alone, which is about that 30% number. And so why is this coronavirus, so different from the others? And it really has to do with this. And let me just orient you to this graph real quick. On the y-axis, you have the fatality rate of different viral infections. And on the x-axis are the number of people that one person who's infected with a virus, the number of people they'll transmit that to. And so the further you are on the right on the x-axis, the more contagious that is, like measles is way out here. And then where you are on the y-axis is how lethal that is. And you can see some of the other things like Ebola, not so infectious, but very fatal. And our seasonal flu, not so fatal, but more than the common cold, and pretty, somewhat infectious. SARS is COVID-2, the COVID-19 virus, is somewhere in this yellow area. We don't know exactly yet, but the fatality rate's probably somewhere around 1%. And the infectivity is somewhere probably between 3 to 5 of the number of people that are infected. And that's often referred to as the R0 value, and maybe we'll talk maybe a bit more about that later. But that's a measure of how many people, how much of a spread there is. And so this is dangerous in that it's pretty contagious and it's more lethal than the average cold. And that's what puts it into such large numbers compared to SARS or MERS or even some of the terrible viruses that you're familiar with. So what have we done so far to address this? Well, unfortunately, there's not too much we've been able to do other than the public health measures that involve social or physical distancing. And so the pandemics are nothing new. there is a playbook for this that is not novel, and it dates back to even 1918, the Spanish flu at that time. We're taking the same response they did then, which is to try to find out who has the virus, isolate them, trace their contacts, and keep people more separated so that they don't spread as much. And so if you just reduce that R0 value to under 1, so every person who's infected transmits it on average to 1 or fewer people, these things go away. If you don't and they're above 1, they stick around and they continue to spread. It's really that simple. So all of our shelter in place effort, all those things are an effort to get that R0 value to be below 1. And we have done that to some degree across the country and across the world, which has led to what is no longer an exponential growth in the rate of rise of cases. Now, different countries have done this to different degrees, and some of the countries in Asia, particularly South Korea, Hong Kong, and Singapore, have very effective testing protocols and isolation and tracking. And they had some of the earliest infections, but were able to bend that curve, flatten that curve early on during their growth phase, and have had fewer deaths in cases. And then other areas like Italy and Spain and in our country, for a variety of reasons, we had a period of time where we weren't able to detect those that might be silently spreading, and we're a little bit late to start to flatten that curve. So we're flattening at a much higher level. So we've flattened at a rate of 1,500 or to 2,000 deaths per day, and it's not going up exponentially, but it's staying at that probably unacceptably high level despite all our measures. And then I'm sure you've heard in the news of various approaches that people are taking, both for testing that we talked about earlier, and we'll talk more about that, but there are a number of what we call PCR-based tests that are quite good now, but are lacking in some of the things we need to get back to work. For drugs and vaccines, remdesivir has been in the news a lot, as well as hydroxychloroquine that our president has talked about a lot. Remdesivir, I think there's good data now, at least, that it shortens the course maybe a bit. It's not a medicine that makes a huge difference, but it does seem to be helpful, and it's worthwhile to have in the fight against the virus to maybe reduce severity and death. But we need better drugs for sure to really combat this. And then I thought I'd just say a few words about the vaccine candidates that are in the news a lot also. And here, what you've heard from Dr. Fauci and many others is that normal vaccine development takes years. I think there are a number of measures that are happening right now that will compress that time frame to perhaps a year or maybe even less if we're very lucky. And part of that approach is that normally one tests a bunch of different vaccines knowing that most of them will fail. Once one works, then you go into production of that, and to scale production to billions of doses, it takes a long time, and then you're able to administer it. And that production phase is a significant lag from when you even know that you're going to have a good vaccine. What I think we've decided to do that's wise is accept that that if we go into production earlier than normal and make 5 or 6 different batches of different viruses, knowing that most, if not all, will actually fail, that's money well spent, even if they don't work, because might save 4 to 6 months of production time. And given the cost to our economy on a per day basis, I think potentially wasting billions of dollars on producing vaccines that won't ultimately work is probably a wise investment and choice. And that's what's going on, which I think is good. Now, traditional vaccine development involves an inactivated or sometimes a modified form of the virus itself. And there's one trial in China from it's called Sinovac that has gone into monkeys, and looks like there's good data of monkeys getting good immunization to that. But I wanted to focus on three more innovative approaches that have never been developed into a vaccine before, that each of them shortens the time frame for development. And so one of those is from a company called Moderna that also has been in the news a lot, I'm sure you heard about. And this is an approach where the company is using what's called messenger RNA, which is sort of the blueprint of the virus for how it would make its own proteins. And that messenger RNA itself is injected into the body, uses our machinery to make the proteins of the virus, and then hopes that our immune system will respond to those viral proteins that are made. So they reported last week that in a phase 1 clinical trial in humans, that at least 8 individuals developed antibodies with that approach. We don't know yet if those antibodies will actually prevent infection, but it's promising. And the time frame of going from the viral sequence to the first test in man was really only 6 to 7 weeks, which is amazing. So if that works in the next phase trials, which will happen by the end of the summer, that is a vaccine that could be scaled to production even before the end of the year and being administered. So that would be a fundamentally different approach to vaccine development. Now, there's another company called Inovio that's developing what's known as a DNA vaccine. And so this is taking the viral genetic material, making it to DNA and injecting that. And the rest of that is very similar to what Moderna's doing, and that's proceeding. And then Oxford Biosciences has been in the, actually it's the University of Oxford has been in the news a lot also for yet a different new way to approach this, which is to actually deliver the viral genome through an adenoviral, another viral vector that's often used in gene therapy. And there also production can be faster than more traditional forms of vaccines. And they too have promising data in non-human primates, and it's moving forward. So I say all that to give you hope that it's a different world right now. And so all the rules of the past, I think, don't necessarily apply to where we are in this current pandemic. Now, with that backdrop of what's going, the landscape of what's going on in the world, let me share with you where Gladstone science is inserting itself and making an impact. So, we've our work is largely in 3 major buckets. It's in the diagnostics, so testing, finding new treatments or drugs, and then in prevention, like a vaccine approach. And I won't go through the work of all of our investigators, but try to highlight a few in those areas. But in fact, at least 10 of our investigators out of 25 have shifted their attention to this virus and lending their varying expertise, even beyond our virologists, in unique ways to try to fight this, which is really remarkable. And I should say the whole community has come together, our administration to support this, our scientists, our core facilities to make all this happen. And I really couldn't be more proud of how they've responded. So let's talk about diagnostics first. And this is a really interesting space where what the country really needs to get back to work is an inexpensive test that can be done frequently, maybe even daily or a few times a week, in workplaces, in schools, maybe at airports, maybe at restaurants, at home, so that people can gain confidence that not only they, but the people around them are not infected, particularly since we know that asymptomatic infection and spreading is a real thing. And so the current tests that are on the market and are being developed are mostly around this technology known as PCR that requires amplification of the viral RNA. And so that requires usually a laboratory-based piece of equipment that can't be done at home or in the field. It's not complicated, but it's a little too complicated to be done, say, like a home pregnancy test or infection test, glucose test. And so it turns out that our group here, led by Melanie Ott and Jennifer Doudna and Dan Fletcher, who's a bioengineer at Berkeley, had been developing such a test exactly for HIV over the last year and a half. And the idea there was that we needed a test that could be done in remote villages in developing countries that were far away from a laboratory. And they had developed a beautiful system where one could use the CRISPR machinery, it's a gene editing machinery, but here to detect viral RNA. And that detection would be coupled to a fluorescent emission of a fluorescent signal that could be detected by the camera in anybody's smartphone. And so by making the smartphone camera function like a microscope that could detect fluorescence, we increased sensitivity to a very high degree and therefore no longer required amplification of the viral genetic material. And that allowed it to be moved away from a laboratory out in the field simply by using a smartphone that even in developing countries is actually now quite ubiquitous. And so we had developed a prototype that I'll show you in a moment for HIV, and we were able to, within weeks, simply by changing what's known as a guide RNA with the sequence of this virus, coronavirus, able to switch out that and make it relevant for the coronavirus that's causing COVID-19. And so let me just show you how this works. We can take a nasal swab, or now testing even saliva. If it has the virus in it, then when we lyse that virus or break it apart and incubate it with this CRISPR machinery, which is basically a protein shown in this blob, along with an RNA strand that it matches the virus of your choice. So this could quickly be used for any virus that comes up now or in the future. And then when all that is put together, a fluorescent signal is emitted. And as I mentioned, the smartphone camera will detect that and tell you if it's positive. Now, because it's detected on your phone, you can design an app to go with that that will in real time aggregate data internationally and with geolocation information and maybe symptomatology. So it'll be extremely valuable for epidemiologic studies and tracking and things of that nature. So this is a very powerful approach that doesn't require amplification, can be done in the home ultimately, and will help with tracking, isolation and tracking and epidemiology. So at the moment, we have a prototype that looks something like this, where it's a handheld device. You slide a phone into it or use a phone that's fixed there. And you just put in a cartridge with your sample and for each patient and pop that out, dispose of that and move on to the next patient. And so we're trying to rapidly develop this in the next few months and get to scale by the fall because this sort of thing, if we can get it down to $5 to $10 per cartridge price, is something that workplaces and schools can use to screen their population. So this is, we're hoping to rapidly scale and compress. Bain Consulting has donated pro bono a group of project managers that are familiar with the space to help us accelerate our time frame, and we're working with a number of other commercial and government agencies to advance this. Now, what about treatment? So if one does get infected with the virus, the first thing the virus has to do is to infect our cell, gain entry into our cell. And it does so by attaching to a couple of receptors that we know on the surface of the cell allow that entry. And so one point of attack for a treatment is to not allow that virus to enter our cells. And Warner Green, who was the founding director of our Virology and Immunology Institute, had years ago developed a cell entry assay for HIV that became commonly used across the world and led to some of the development of solutions for HIV. And so we've quickly adapted that assay to screen for drugs to prevent the entry of this coronavirus. And so Warner is at the moment gearing up to screen a library of about 12,000 drugs that have already been through some safety test through the FDA or other agencies around the world. And so if any of those are actually prevent entry, the path to clinical use is very quick because they're already tested in early phase trials, they're safe in humans, and they go directly to an efficacy test. These 12,000 drugs actually were put together in part by one of our Gladstone investigators, Sheng Ding, who is a chemist and was recruited back to China a few years ago to lead the Tsinghua New School of Pharmacy, which is one of the premier universities in China. And they, together with the Gates Foundation, have put together this panel of drugs for exactly these sorts of uses. And so we have developed a new facility at Gladstone, a high safety level dedicated to growing live virus. In that facility, we're set up now to do this screen with this drug library. So stay tuned for that. That's will, I think, hopefully yield some very interesting results. Now, once the virus enters the cell, it has only one job, and that is to make more copies of itself and spread. And a virus is really a bag of genetic material. It has absolutely nothing else. And so the only way it can do that and make more copies of itself is by hijacking our own cell's machinery to make more of itself and then package itself and spread to the next cell and the next individual. And the way it does that is that it makes its own proteins. There are less than 30 proteins that the virus itself encodes for. And those proteins then have to work with our proteins to do its thing. And so Nevin Krogan and Melanie had developed a platform several years ago in our what we called our BioFulcrum program that was supported with some generous philanthropy to create a platform to use the state-of-the-art proteomics or mass spectrometry equipment that we had collected at Gladstone over the years and map all of the host, which in this case would be our human proteins that interact with various viral proteins, because those would serve as the drug targets. If you disrupt that interaction of the virus proteins with our proteins, then it can't do its thing. It can't make more of itself. And so they had been doing that for the flu in collaboration with a biotech company called Vir here in San Francisco, and for several other diseases. So Nevin quickly took that exact same platform, and within a matter of weeks after the viral genome was sequenced, made all the viral proteins, put them into cells, figured out all the human proteins that they interact with. And we reported that just 2 weeks ago in the journal Nature. It got a lot of press across the world. You might have read about this, but it created the first roadmap of what proteins this coronavirus interacts with in our bodies to take over our cells. And that map was quite complicated. It looked something like this. And each of these red diamonds represents one of the 26 or 27 viral proteins. And the spokes around that are the human proteins that it actually binds to and interacts with. And what Nevin found is that, in fact, this virus is working through so many different pathways in our cells, which probably is why it affects so many different organs. As we learn more and more about this virus, it's affecting not just the lungs, but the heart, the kidney, the brain, and our immune system. And so this is the roadmap basically in front of you that we're able to discover in a matter of less than 3 months, which is record time. And this was through a broad collaboration with scientists all over the world. And they took it one step further and actually took some of these where there were known drug candidates that actually had already been known to disrupt those interactions. And many of those now are advancing already to clinical trials because they're already proven to be safe. And many others we're now testing that haven't been tested in our system with human cells to see if they disrupts the virus's ability to make more copies of itself. So this right here, what you're seeing, is going to serve as the world's roadmap for how to find new drugs for this virus. Now, those are some of the areas where we're trying to make new drugs, and they're very promising. But what about vaccine development? So Lior Weinberger, one of our other really creative investigators, had developed a very novel approach for an HIV vaccine over the last several years that the Department of Defense had been funding significantly because they have been worried about such pandemic spreading, and the approach Lior developed for HIV, they knew could quickly be adapted for any future virus pandemic. And the clever part of how he's approaching this, which is very different from all the other vaccine approaches I mentioned to you earlier, is sort of turning the tables, if you will, on the virus. The virus normally hijacks our cells, as I mentioned. The approach Lior has developed basically hijacks the virus's strengths. And the way it does that is that for HIV, he created a mutant form of the virus where he took out many of the genes the virus needs to cause trouble and cause disease and do bad things, but he kept the parts of the virus that allow itself to replicate and package itself and spread. But if you find the right combination, you could basically be making a mimic of the virus that is completely ineffective in causing any trouble, but it does make copies of itself even better than the real virus, and essentially outcompetes the real virus to spread, and that serves as protection against the real virus. So he coined the term therapeutic interfering particles because they're particles of the virus, but they're therapeutic because they interfere with the normal virus's function. But essentially, it allows these mimics can make more of themselves, package themselves, and spread to the next cell. And by doing so, they confer immunity, confer protection. And so he had, before this pandemic, proven up that approach for HIV infection in mice. And so the US Army had come to him and said, we want you to do this now for the same thing for the coronavirus. And so he's doing that at the moment and testing the right combination of viral particles to leave. And once we do it in tissue culture in the next few weeks, then it'll be taken into mice in our facility here at Gladstone. And if it works in mice, the government is saying right now that we would bypass any further testing and go directly into a phase 1 trial, shooting for being able to do that in the fall also. Now, one of the beauties of this approach that also is could be a complication also is that this approach not only spreads cell to cell through the virus, but potentially could spread from person to person, conferring broader immunity without having to dose everybody. And so when you think about the challenge of making 7 billion vaccines, even if when we find something that works, that's an enormous challenge. And you're already hearing in the news about nationalism playing a role in which countries will be able to make and get the virus and administer them first. If this approach works and it even spreads person to person, then you can imagine we'll be able to create herd immunity at a much faster pace. Now, so that's what an approach we're taking for vaccines. I wanted to touch on one other area, which is the fact that when people get infected with this virus, there are 2 organs in particular that are affected that are causing this high degree of lethality. And those are the lungs and the heart. And in both of those, it appears there's some infection of the tissue, but another big component is our immune system overreacts. And the immune system's overreaction itself makes the problem even worse. And that's particularly true in the lungs, likely in the heart as well, where that's probably why we hear of the stories where people get infected, they're sick for a week or 2, maybe they're getting better, and all of a sudden they get worse quickly and end up on a ventilator. A big part of that is probably the immune system attacking the lung. And so Melanie and Warner, Melanie Ott and Warner Green are working on trying to figure out what is that immune response and how could you modulate that, specifically with human lung cells. And so, as many of you know, we've had a world-class stem cell program at Gladstone for many years. And through that, Melanie had already been developing these stem cell-derived lung organoids. So they formed a three-dimensional architecture of the lung from stem cells, and with multiple different cell types, including inflammatory cells. And so we're using that and new single-cell approaches to dissect how the immune system is overreacting, how we might be able to intervene in that. And Todd McDevitt's lab had been working on how the immune system changes as you get older. Since we know older people are more significantly affected, we're able to study that aspect in this model as well. And then with our cardiovascular efforts here, particularly in the stem cell field, Bruce Conklin, Todd McDevitt, and Nevin together are working to take human stem cell derived beating heart cells that many of you have seen in our labs, and figure out what are all the proteins that the viral proteins interact with in a human heart cell to take it over to try to find new drug targets that would affect the pathology specifically that's going on in the heart. And so I haven't given you, shared with you all the things that our investigators are doing, but I've tried to give you a flavor for the breadth of what we've been able to bring to the table very quickly, just in a few months, to try to tackle this virus and really make a difference because of our unique expertise. There are certain areas that I mentioned where because of our existing efforts, we clearly had an advantage to try to make a difference. And so we've tried to do that as part of our social and scientific responsibility. But I can tell you that all of our investigators and scientists in the lab feel like, given where we sit in the landscape, we have an enormous responsibility and part of our mission to step up to the plate during this time and help the world in ways that we are uniquely situated to do. And I think you can tell from what I described, we are in fact doing that. And many of the things I described will not only be applicable to this virus, but are being set in place for even the next pandemic, because this won't be the last. If history has taught us anything in this century alone, we'll get through this one, but there will be another. And the more infrastructure we can build during this time, we'll be better off the next time. And so that I'll leave you with just the notion that where an institution like ours can really make a difference is in doing the basic science to discover the drug targets, testing those preclinically, leading to clinical trials, anticipate the future, and really get at mechanism. Because once you understand mechanism, that's how you'll enter into new approaches. So with that, I thank you for your attention, and I'll open it up to some questions, and we can have a discussion about these things. Thank you.
Thank you, Deepak. If any of you has a question, just click on that Q&A button at the bottom of your screen and type in your question. And I'll try to read them or collate them for Deepak. Deepak, there's one question about privacy issues with using a cell phone. And do you anticipate, for diagnostics, do you anticipate any problems there?
It's a very important issue, and it is key that the data that is then collated and aggregated is HIPAA compliant, which is a term people use for various regulations, so that it's de-identified. And so the data that would be on there would be positive or negative and would correlate with some other data like symptoms and geolocation, but not be correlated with a name. And so the privacy concern is extremely important and would be in place.
Someone asked, did any of the 8 humans that were given the trial drug have side effects? That must be the Moderna.
So this is the Moderna. So they were a broader number that were given the trial, about 45, I think, in that trial. And many of them developed some types of antibodies, but there's one type of antibody called neutralizing antibodies that are specifically the type that might prevent that viral entry. And 8 of them developed that. They did not have significant side effects in that study, which is a very important thing because vaccines can often be derailed because of various side effects. And because this is such a new vaccine approach that's never been tried, that itself was a very significant finding that there weren't bad things.
Good. How about what role, if any, do cell surface coreceptors, in addition to ACE2, have in the SARS-CoV-2 infection?
Yeah, so this question gets to, it's been widely reported that there's a receptor called ACE2 that is a main receptor that allows entry to the cell, but there is a second receptor that is a co-receptor that works with it. And so in all the studies we're doing, we're looking at both of those to see if we could inhibit either one of them to limit entry. So that does play an important role. And then I did mention that there's a protein on the surface of the virus called a spike protein, which gives its name of coronaviruses. They all have these spike proteins that is like corona stands for crown, that creates this crown-like appearance. And it's that spike protein that we think interacts with these receptors, so that many of the vaccines are in fact using that spike protein as the antigen or to raise immunity against, because they think that would then bind to it and prevent it from interacting with the receptor.
Thank you. Elementary question, but what is it about bats that there are so many viruses coming from them?
Yeah, you know, bats carry over 100 different coronaviruses that they live with. So they have been, an important sort of vector for the animal to human jump of many of these coronaviruses. Why so many survive without causing bats trouble probably has something to do with their immunity, but I think it's unclear at this time, but they are the biggest source because there are over 100 different ones, which is why I say that it's unrealistic to think that there won't be another one of these. It may be more like SARS or MERS that was less contagious, so we were able to make it die out on its own by public health measures. But it could be even worse than this one, so we have to be prepared for the next one.
So there are a number of questions on how do you prioritize funding these various projects, and which ones are looking for more philanthropic support?
Yeah, that's a great question. I should say that because the normal funding of science is through government grants, we've applied for a number of grants for these various projects, but none of those, in fact, because of the time it takes for those to go through review, have started. So all the work I described to you, we've been doing bootstrapping with funds that people are either repurposing or through new philanthropy, which I'm very grateful to many of you on this call today who've generously stepped up to support this work in this time of crisis and partner with us. And so we do each of these projects. We have written up a white paper that's short to describe and lay out what we need to accelerate them. So we are raising philanthropic dollars for each one of these. How to prioritize them is a bit tricky. They're all actually really important. I can give you an example. The Roddenberry Foundation, who supports our stem cell center, recently said that they wanted to support this as well, and they gave us a few $100,000 with the notion that depending on what panned out from that, they would support a bit more. And we actually are going to take those dollars and spread them among 5 or 6 projects evenly as sort of seed dollars to get the first initial experiments done, and then see which is most promising, and then double down on those that are most promising in a second tranche. So something like that. I think we want to take the most creative approaches right now, and sometimes those are riskier, but I think that's exactly what we need to do. So I would welcome any of you who want to make a difference in this pandemic and partner with us. We would love to do that.
Thank you. GlaxoSmithKline recently announced a collaboration with Jennifer Doudna on a CRISPR-based mobile diagnostic for COVID-19. Is this the same or related to the one you just described?
Yeah, that's a great question. It's related. So Jennifer had co-founded a company called Mammoth Biosciences several years ago, and they are focused on using a CRISPR-based platform for a variety of things, including diagnostics. And so they have developed a diagnostic that uses CRISPR-based approaches to detect the virus, but that technology right now still requires what we call reverse transcription and some amplification, essentially, of the viral RNA. And so the approach is similar, but requires this additional step that makes it so that it still requires a laboratory base. And so we are working together, given Jennifer's connection with the project I described and with Mammoth, we're working together with Mammoth to take our technology and advance it towards what they, in a way that they actually described in their collaboration with GlaxoSmithKline, which is the desired outcome, which is to ultimately be able to have a mobile test. Because we are developing, as I mentioned, this for HIV, we have a prototype already. And so we're working together with them to see how we can advance that in a way that could get out by the fall.
Great. Here's one on my mind also, but another person. What work is being done to understand whether antibodies to this coronavirus confer immunity? For how long and at what level is it needed for immunity?
Yeah, so that's another great question. So you've heard a lot about antibody testing. And so there are a number of antibody tests. And in fact, Alex Marson's lab reported recently tested about 13 or 14 different antibody tests that are on the market right now to see how good each one is. And some are better than others. But what we don't know right now is how much immunity, if any, do those antibodies confer and how long, and if you can get reinfected. For most other viruses, these neutralizing antibodies do confer some protection. So it'd be a surprise if that didn't happen here. What we don't know is, does it completely prevent you from getting disease again? Or does it, if you got infected, would it kick your immune system in earlier and you would have a much more milder form? There have been tests of people who tested positive or negative, then tested positive again, which might suggest that you could get reinfected, but maybe don't have a severe disease. But I think the jury's still out. It's too early to know the definitive answers to those questions. But it's an important one because both convalescent plasma, so plasma from somebody who's been infected but raised antibodies for other viruses like Ebola, had been proved to be an effective therapy to transfuse that into somebody else with the infection. And so that may be the case here, and there have been clinical trials with that already, and it looks like it helps. But that would probably only last a few weeks, is what we think, maybe 2 to 3 weeks. So all that is still in play and being investigated right now. So there aren't definitive answers to those, but it's an important area of study.
Okay.
And in our, I should say, in our BSL-3 facility, we can infect human cells with the virus and test some of these antibodies in plasma to try to answer those questions in terms of the degree of prevention of viral entry.
Do you have any insight into the incidence of Hashimotos in children with COVID in New York City?
Yeah, so there's this inflammatory, body-wide inflammatory condition that we're starting to recognize occurs in children and young adults. It's been prominent in New York. Washington, DC Children's Hospital has reported almost 150 kids with this. It's a body-wide inflammatory condition that's actually most similar to Kawasaki's, not Hashimoto's, Kawasaki's syndrome. And I'm quite familiar with this because as part of that syndrome, the vasculature in the body gets inflamed, and in the heart, the coronary arteries develop these aneurysms that vessels get dilated and are at risk of rupture. So it's a common thing we see in kids with heart disease. that develop heart disease from this. It's very concerning. Fortunately, it's rare, but we are seeing hundreds of cases of this, and it is likely in part of the overreaction of the immune system to the virus. We have seen this with other viruses, post-viral infection syndrome, where it's an autoimmune reaction, and we've treated it before with immunoglobulins, so antibody sort of immune therapies. So that's probably what's going on here, but it's very, very concerning, and hopefully it doesn't grow, but it may be growing.
Okay. Are you aware of any coordinated efforts around outpatient trials for therapeutics?
So some of the initial trials for therapeutics were done in the most severe patients, so those on ventilators, like remdesivir was first tried on ventilated patients. But in fact, it might be more effective early in the disease, and many of these things that might prevent viral entry and replication are probably going to be most effective early in the disease, so more in the outpatient setting. And so there are trials with remdesivir at an earlier stage. There are trials with hydroxychloroquine at an earlier stage. I think there's good evidence that at a later stage, hydroxychloroquine is not helpful. And so yes, short answer is yes, there are trials as outpatients, but we need more things to try than right now. There are things that have already been developed for other indications are being tried. The next generation of clinical trials will be through new discovery specifically for this virus.
So this is a broad question, but how do we prevent nationalism in vaccine distribution?
Yeah, I touched on that briefly because I am very concerned about this, because no matter what works, and even if 3 different things work, there's no way we'll be able to produce enough vaccines to at the same time meet the global need. So how do you allocate vaccines? It's going to cause, I can just tell you, it's going to cause a significant issue worldwide. And there's not a good answer to that. I think the wealthier countries are going to get them first. Our government already contracted with AstraZeneca, who's partnering with Moderna to make this mRNA vaccine. 2 days ago, we signed a $1.2 billion contract with AstraZeneca to help start making production of the vaccine even before we know that it might whether it's going to work or not. So we're putting those dollars at risk because it may not work. But if it does work, then by virtue of being in a position to invest in that early, we are going to get that first. So I think money is going to dictate that in significant part. But you can imagine countries where public health measures are going to be really challenging, where hygiene is just not even possible. And I'm thinking specifically now a country like India that I'm familiar with. For a good chunk of the population, just getting access to soap is not realistic, and running water is not realistic. So the potential for an explosion in a country like that would affect the whole world. If it got out of hand in India, it's not going to stay out of hand there. It's going to spread throughout the world like we've never seen before. China was able to contain that quickly. I don't see how India could do that with their resources and structure. And so one could make the argument that for the benefit of the world and safety, even in the wealthier countries, it's in their interest to make sure the countries that are least capable of public health measures do so. Now, having said all that, India locked down very quickly when there are less than 100 cases. And so they have quickly flattened the curve at a low level. But I'm still very worried that could explode any day.
Right. Here's an interesting question, Deepak. What role, if any, will disease tolerance signatures of asymptomatic carriers have in helping develop treatment protocols? How does science develop a bank of these signatures?
It's a great question. And in every disease, we always look to those extremes where people should have disease but don't, because something about their bodies are protecting them. And if we could unlock that secret, we could use it to our advantage to create therapies. And so I think figuring out why people don't get infected and don't get disease is really important. And so part of that is probably our immune system. And this is a case where I know Alex Marson's group is very interested in those kind of questions, where you come at from the immunology side and ask, what is our immune system doing in those cases? What are those signatures? And can we replicate that? So it's a great approach, and one that we will be pursuing. We can make tailored, say, lung organoids and cardiac cells from the people who get infected but don't get those problems, and actually use their cells through our stem cell world, use their own personalized cells to ask many of those questions.
Great. Can you comment on why New York and San Francisco's experiences have been so different?
Yeah, so this goes back to the notion that in these pandemics, there's usually an exponential growth phase of infections where before we sheltered in place, the doubling time for the virus infections was every 3 days. So every 3 days, the number of people infected and as a correlated who would die would double. And so when it's doubling from 10 to 20 or 10,000 to 20,000, it's not that big a deal. But if you extrapolate that exponentially, then within a month, one goes from 10,000 infections through 10 doubling times, it becomes 10 million. And then from 10 million in 3 days, it would be 20 million. 3 days more, it would be 40 million. So now you go from 10 to 40 just in 6 days, right? And in San Francisco, when they shut down, had the same number of deaths that New York did on March 16th. New York shut down 6 days later, just 2 doubling times later. That probably made all the difference, because we're already at that growth phase where we're at a very high baseline, so each doubling time was enormous. And that probably made the biggest difference, because we went on completely different trajectories just because, in large part, those 6 days. And that happened also in 1918 with the Spanish flu in St. Louis and Philadelphia. There's a lot written about those two cities. They went through the exact same time frame difference of 6 days and had vastly different courses of overwhelming the medical system, death, et cetera. So, our leadership here in the Bay Area really needs to be commended for acting early. It made all the difference in the world, those 6 days.
Well, we want to be respectful of everyone's time. Thank you for those great questions. And Deepak, thank you for answering them. I just want to say again, thank you for all of your interest in COVID-19 research. As you've heard, the Gladstone Institutes have been able to move very quickly along a number of fronts because of generous donors. If you wish to learn more about how you can accelerate progress, contact us or go to the Gladstone website, which we've put in the chat section of this call. We welcome your partnership. We wish you a good day and a great Memorial Day weekend. Thanks so much.
Bye, everybody. Take care.